Thank you. Well, normally at this point I would thank the organisers for giving me the opportunity to speak at today's conference, but seeing as I'm also a member of the organising committee, I can't quite bring myself to do that. Hi, my name is Chris Proudfoot. I'm a postdoc in Bruce Whitelaw's group, and I'm going to talk to you today about some of the projects we've been involved with genome engineering livestock species. To start with, Dollys Legacy. So what is Dollys Legacy? Well, some people would say it's an animal rescue centre in Lincolnshire, but I suspect that's not the answer that people are looking for. But Dolly, the world's most famous sheep, who got herself onto the front cover of Time magazine, at the time was thought to have given us the ability to induce precise insertions and deletions easily and efficiently in the germline of livestock species. And as it turns out, and as Ian touched on, it's not as easy nor as efficient as we once had hoped. Now, whereas the rodent communities have stem cells that have allowed them to bring about increases in efficiency in this technology, the same is not the case for livestock species. We don't have stem cells that we can turn to. And so as a result, the livestock community has had to look elsewhere for this improvement in genome modification efficiency. And ironically, the first of the tools that the community would turn to was first described in the same year that Dolly was born, and this was the first zinc finger nuclease. This is the first of the genome editors. And as Goetz has already mentioned, there are a number of other genome editors that have appeared in the scene, namely the Talons back in 2010, and more recently the CRISPR-Cas system and more recently in 2012. And what these allow you to do is make very precise modifications of your genome of interest at far greater efficiencies than have ever been possible before. And Roslin, here at Roslin, we have experience of using all three of these genome editors. Genome editors can be applied in a number of different ways. However, their predominant use is the generation of a double-strand break at a very precise locus in the genome. And unfortunately, Goetz has already explained this slide, so I'll rattle through it quickly. If you leave the cell to its own devices, it repairs the break through non-homologous end joining, which results in indel formation and disruption of your target locus. You can provide a template for repair utilizing the homology-dependent repair pathway, and if you're wanting to make a small change, you would provide a single-stranded oligo, and if you wanted to make slightly more complex changes, you would provide it as a double-strand plasmid, and this would lead to modification of your target locus. And finally, the last modification you can make is you can delete regions of the genome, where you would design a genome editor that would flank your region of interest. Non-homologous end joining would then repair the breaks, deleting the intervening sequence. And we have examples of animals that we've generated that contain all three of these different modifications. In terms of genome editing in livestock species, most of the work that's carried out involves the technology that was pioneered by the Dolly team. In this case, you would generate your edit of interest in a primary cell, followed by nuclear transfer to generate your edited animal. And as they do ag research, here at Roslin, we prefer a slightly different approach, where we directly inject our reagents into the livestock into the zygote of livestock species before transferring to a recipient animal. And I'm just going to take you through a few examples of the animals that have been generated using this methodology. And the first being gene disruption, and that was carried out in the myostatin gene. Myostatin is a negative regulator of muscle growth, and the Belgian blue cattle have a deletion in this myostatin gene, which leads to loss of functionality of the protein, promoting what's known as the double muscle phenotype, resulting in this beast here. This all comes about from an 11 base pair deletion in exon 3 of the gene, which leads to a frameshift, bringing into frame a downstream stop codon. This results in premature truncation of the protein, which results in its loss of functionality. And so our aim was to introduce a similar mutation into the genome of sheep species and cattle that didn't naturally have this mutation. At this point, I'd like to point out that this work was carried out in collaboration with Scott Fahrenkrug and Dan Carlson at Recombinetics and Chuck Long at Texas A&M. So Recombinetics produced the talon pair that would generate a double-strand break just downstream of the equivalent sequence that was deleted in the Belgian blue cattle. And we had 2 animals born using these reagents. The first is a wild-type heifer, and the second is a mosaic bull, which has a wild-type allele, a delta arginine 283, and a delta 1. And in this case, the delta 1 leads to a truncation of the protein very similar to what is observed in the Belgian blue cattle. And at this point, I should point out that analysis showed this allele to be the predominant allele in the animal. Unfortunately, we lost 2 edited bulls at calving due to complications unrelated to the editing, but that essentially gave us an editing rate of 75%. When we carried out the same experiment in the sheep, we used the same talon pair, despite there being a SNP in one of the talon binding sites, as it is well published, well documented, that talons can cope with a single base pair mismatch. We had 1 animal born that was edited, and unfortunately it was only a delta arginine 283. And unfortunately there was also a hit in editing rate, where we only ended up with 7.5% of the animals born were edited, and that perhaps is a result of this SNP here. When you look at the animals shortly after birth, there's no obvious phenotype in the cattle. We wouldn't expect one in the lambs, but if you fast forward 6 months, there's a clearly increased muscle architecture in the bull on the right compared to the heifer on the left. We can also use this technology to generate modifications, and this was done when attempting to generate a sheep model of a human neurological disorder. And this specific neurological disorder is caused by a single amino acid change, and in this case we used the CRISPR-Cas system to introduce the desired change. So we were able to design a guide RNA that would target the Cas9 to generate a double-strand break just downstream of the codon we wanted to change, and then provided a template for repair which contained the sequence we wanted inserted at the break site. Incorporation of this sequence at the site of repair also acts to prevent the recutting of the repaired allele. We had great success using these reagents, where nearly 50% of the animals born showed editing events, and 13% of live-born animals were homozygous for the desired change. An example of a larger modification at the target site can be seen in our African swine fever project. African swine fever virus is endemic to sub-Saharan Africa, and it exists through a natural cycle of infection between ticks and wild pigs, such as bush pigs and warthogs. Now, whereas the warthogs show persistent infection with the virus, they show no clinical signs of the disease, whereas the domestic pigs suffer a hemorrhagic fever with an extremely high mortality rate. So it posed the question of, is there a gene responsible for this key difference in immune response? So genome database searches were carried out for a potential candidate gene, and one that came out of the mix was Rel-A. Rel-A is also known as P65. It's one half of the NF-kappa B transcription factor, and one of its roles is involved in the regulation of immune response. If we take a look at the sequence of the transactimation domain, there are only 3 differences between the warthog and domestic pig sequences. One of them is 513, which in mice and humans is shown to be a phosphorylation site. You have 448 and 485, which are proposed to be phosphorylation sites, but it appears that warthogs have a less active form of the Rel-A. So it posed the question, is this the reason for their resilience to ASF infection? And if we then introduce these residue changes into a domestic pig species, would we make it resilient to ASF infection? So this work was carried out in collaboration with Fyodor Urnov and Ed Rebar at Sangamo Biosciences, who produced a zinc finger pair that would generate a double-strand break in the genome just upstream of the first of the residues that we wanted to change. We would provide a template for repair, and this time as a supercoiled plasmid containing all three of the residues we wanted to change, and it would also contain an extra silent blocking mutation to prevent the zinc fingers recutting the repaired allele. And so essentially what we were looking to do was change 5 base pairs, but in this situation, in this case, spread out over a region of 262 base pairs. This would then be flanked by 600 and 974 base pairs of homology. We ended up with 3 edited animals born, which accounts for 7% of all live-born animals. The first was pig 364, which was shown to be heterozygous for the warthog change at all 3 positions. Pig 367, which was homozygous for the first 2 changes, but retained the pig residue at the 3rd site. And most successfully was pig 563, which was shown to be homologous for all 3 changes. So the position we're at now is breeding up a cohort of these animals to assess the question, are they now resilient to ASF infection? And the last example is of a deletion, and this was carried out on a cell surface receptor protein, where we wanted to delete a domain from that protein. This domain to be removed is coded by a single exon. So it was simply a matter of designing CRISPR-Cas reagents that would generate a double-strand break in the intronic sequence that flanked this exon to be removed. And using these reagents, we generated 3 edited animals, which is 50% of the litter. On 344, you can see that we have a wild-type animal. 346 is heterozygous for the deletion, and 345 and 347 are homozygous for the deletion. You may notice that there's a slight difference in the size of the deleted product, and that's a result of the indel formation at the site of repair of the deletion event. Functional analysis of this protein showed that the protein is still expressed, it's still present on the cell surface, and it also retains functionality associated with the remaining domains. So just to summarize the editing events that I've discussed during this talk, Genome Editors are a hugely adaptable tool that allow us to generate modifications in livestock species with very high efficiencies. Whether you're looking for a small gene disruption or a larger gene modification, we have this ability now to do this in livestock species. Admittedly, some would say that 7% is not a particularly high success rate, but I would also point out that ultimately this technology is still in its infancy. Efficiencies are improving, and they're improving rapidly. So come back in 3 years' time, I guarantee that number will be somewhat different. But in terms of a group carrying out livestock research, that's made a big difference for us. Our group historically has been focused around disease resistance and resilience, whether it be PRRS, ASFE or influenza. But it's now meant with the ease that we can now generate edited animals, it's opened up a whole number of different research areas to us, whether it be generating models of human disease, going down the bioreactor route, producing proteins in milk or eggs, and obviously production traits, whether it be fecundity or meat or milk quantity and quality. And ultimately, with the advent of genome editors, we're now at a position where, in terms of generating genome-modified livestock, we're now at a position where we're limited more by the imagination of the researcher and less so by the technology available to carry out these modifications. With that, I would just like to finish with a few acknowledgements. Bruce and his group, it's a fantastic team to work with. Alan and Christine, who have been involved in a lot of the projects we work on. Genus, who are major funders of a lot of the work our group carries out. Our collaborators, Recombinetics and Sangamo Biosciences. And obviously with all large animal projects, we would be nowhere if it wasn't for the fact that behind us is a fantastic farm staff. With that, I would like to thank you for your attention, and I'll take any questions.
Other questions for Chris?
Yes. So the ASF experiment is a lovely one. It's a great experiment. So assuming that this resilient to the ASF is transmitted to the pig, how likely do you think that pig farmers will pick up on this and will accept using this pig for changing all the animals they will be producing?
As with every genome-modified animal, there will always be some resistance to large-scale dissemination of the new genetics. But I suspect that as we have now with ASFE creeping into Eastern Europe and it's slowly impacting on European pig farming stocks, I imagine that their opinions may change in the near future. But I suspect that it should be quite a positive one. Certainly the interactions we've had with the farming environment has been a very positive one. They like the idea of there being a way that you can tackle these diseases, especially when you speak to them about FMDV. I mean, it's still a very sore subject in Britain. And my father-in-law was unfortunately one of the first farmers in Scotland to suffer from an outbreak. And he thinks this sort of thing is fantastic and that he's all for it.
Other questions for Chris?
I was curious, how do you deliver the zinc fingers or Cas9 proteins in the diet to the zygotes?
So most of the time we would deliver them as mRNA. Back in the very early days, they were delivered on plasmids and expressed from plasmids. But we realized very quickly that led to sort of very high levels of mosaicism in the animals. And so ultimately, when you're delivering at the one cell stage, you want your reagents to go in, generate your editing event, and then go before the cell divides, so that you have your animal with a single editing event rather than multiple different alleles. So we're now at a point where for all this work, it was delivered as an mRNA. With the Cas9, now what a lot of people are doing, and certainly it's very popular in the mouse community, and we're investigating it in the livestock community, is delivering the Cas9 guide RNAs as a pre-complex. And so it's almost like injecting a restriction enzyme. It's good to go as soon as it's in the cell. There's no translation required. It can generate its editing event and then be degraded.
Any other question? Not at the moment, okay.
Sorry to rain on the parade slightly. What evidence do you have that it was a single allele that imparted the protection against ASF? And if it is polygenic, then how many alleles do you think you could actually knock out or edit?
Well, unfortunately, I'm not an expert in the neurological condition that they were targeting, but our collaborator who we're working with, he works in the institute, was ASF. Right, okay. Very.
65. I presume there was some bioinformatic evidence in the background, but I could imagine it being polygenic, even between warthog and pigs.
Yeah, there's a very high chance that it will be, but we are starting off with our best candidate, asking that question, and then we have animals that we can add on subsequent edits, should that be the case.
Okay, we'll stop there and carry on. Thank you very much, Chris.